Good morning, everyone. Welcome to Science 200, week number 2. So you've already made it through your first week. You worked on your discussion board postings. You did some work in your textbook. You learned about the scientific method. So let's take a look at what we've got going on this week. A review of last week very quickly. What are the natural sciences? So you went through those and you figured out which one you were more interested in. Course, if it were me, it would be biology. But some of you said astronomy, which is also very cool. Environmental science. I love environmental science. I was an environmental scientist for 11 years when I worked in vector control. So I love that too, and I love all of nature. But there's lots of different environmental sciences, physics and chemistry, etc. So we took a look at what you were interested in through the discussion board postings, and hopefully you picked a subject for your research this term having to do with the one that you were most interested in. You learned how science works. It is a process. It is not step 1, step 2, step 3. That is not how science works. It's a process. And you have to do things over and over and over again. You have to have peer review in science to keep everyone honest and to figure out if there are problems, if there's things that you didn't think about when you did your experiment and you came to your conclusion. Was there something that you missed? Was there something that you did wrong? Was there something that you did right that you figured out that people are really excited about? So how does science work? You looked at that and the importance of revision, because revision is extremely important. Oh my goodness. When I've written up my research, I can't even tell you how many times I wrote it and rewrote it, and I would let somebody read it and they would make changes and I would fix it, and I'd let somebody else read it and they'd have other changes, and in my whole research group, everyone would read my work and then they would suggest changes or have questions about it. Then you send in the work to a journal, and the editors there and your peers who reviewed your work, they'll have suggestions about it, which you have to go back and do revisions on. So revision is a really important part of science because other people have eyes on your work. And of course, I'll be having eyes on your work. And you picked out your topic for the term, whether it's electric cars or genetically modified organisms, ocean acidification. We had all different types of topics that you could choose from. And you could even choose from other ones, but you definitely have to run those by me if you want to choose something different. Just to make sure, I will make sure that you are able to do this. All right, so what do you have this week? This week is very intensive in the textbook. There's a lot for you to read and a lot of questions to go through. By the way, that's dolphin strand feeding down there in that picture, which is a really cool behavior that dolphins in my area have. So anyway, this is chapter 2, and it's going to take you through the process of science and how it has worked in 2 different examples. One is Alexander Fleming. Now, y'all, I'm a microbiologist, so this story is near and dear to my heart. Alexander Fleming was a microbiologist. He ran a lab. He came in after vacation and was cleaning up his petri dishes, and he noticed one. He'd been growing bacteria on these plates, and he noticed one that had some mold growing on it. And that wasn't the important thing. The important thing was that there was a very clear zone all the way around that mold where no bacteria had grown. They'd grown right up to that zone, but they didn't pass over it. And he said, there must be something going on here. That mold must be making some kind of chemical that's inhibiting the growth of these bacteria over there. And so he started examining this, and he isolated a chemical compound that he called penicillin. It was named after the type of mold that was growing on his plate, Penicillium notatum. So he isolated this chemical, and he was finished with the work. That's as much as he was interested in. So the textbook takes you through what else happened then, because all of you have heard of penicillin, so you know that penicillin is a really important antibiotic. Well, if Alexander Fleming had stopped at that point with his examination of penicillin, and he didn't think about how you could possibly use it or what you could do to make it, we would not have antibiotics today. So it took other scientists moving through this process of taking this, growing it better, finding a new kind of mold that would grow it better, making lots of mutations in the mold so it would make lots and lots more penicillin, and then producing it on a grand scale before anybody could actually use it. And it was used widely in World War II by the United States troops. It saved many, many lives during that war. Because one thing that happened in wartime was that you developed infections caused by wounds, shrapnel, that sort of thing, or even from the surgeries that you had to have. So penicillin saved a lot of lives in World War II. And then, of course, we started using that in the United States and across the world after World War II. So that's a really important story in science. And then there's also, I will say, a very long story about John Snow and how he showed the world that cholera, which is a disease of dysentery, so it's a type of diarrhea, and it produces something called rice water stool, where just water just flushes out of your body. And it's caused by bacteria. They didn't know that back then in the 1800s. We still did not understand that back then, that there were tiny microbes that could do something like this to you. And so, hang on. Okay, pick back up. So John Snow figured out that cholera was being transmitted between people through water that was contaminated by feces from other people who had cholera. This was not the preferred thinking back then in science. There was something called miasma, and this was just bad vapors in the air. So you breathed in these bad vapors and you became sick. And so most people thought that diseases were transmitted through miasma. Now we know that some diseases are transmitted through aerosol droplets. So this is true. If you're thinking about something like tuberculosis or cold or COVID, the flu, there are lots of diseases that are transmitted through aerosol droplets. Cholera is not one, and not every disease is transmitted through bad vapors. Okay, so John Snow took a look at this, and he went to extremely great lengths to show that cholera was actually transmitted by contaminated water sources. And so you're going to read all about it. Now, I will tell you that no scientist on this planet wrote that textbook passage about John Snow. It is very long. It's a lot of pages. So you're going to have a lot of work to do in the textbook this week. I'm just saying. It's very interesting. I'm not giving anything, I'm not saying anything bad about the textbook, but it is long. And you're going to see how science works and how John Snow's work really, really helped advance public health. And public health is being concerned about the health of the whole public instead of just one individual. And this is really important in transmission of diseases. You know how that works. You've watched it in action with COVID, right? Why did we wear masks? Why was there a mandate to wear masks? So that we wouldn't transmit disease to someone else. Perfectly fine if you get the disease and you don't care about it, but you cannot transmit it to somebody else, and especially on purpose. So that's how public health works. And you're going to learn how science works through these two examples that they give you. Your topic this week, you are going to figure out a research question, some kind of question that you want to answer or you want to find the answer to that is already out there. And you're going to have to find it because you can't do experiments carry out experiments to do this, to figure out the answer. So you're going to ask a really good research question about the topic that you have chosen, and I will be grading your issues and significance sections this week, starting tomorrow, Monday. So you're going to figure out a research question to ask, and be watching for my grading of that when I get that far next week, because a lot of students have to revise this because they're usually not, well, not usually, but sometimes they're not answerable questions. You know what I mean? We have to look for something where you can find a particular answer. So work on your research question. And then finally, hang on, let me get to the next slide. Okay, finally, my tip of the week for you is to break up your assignments into manageable pieces. You're going to have a lot of reading to do this week, and no other week will be like this one. I'll point that out to you. Break up your assignments into manageable pieces. So you know in your textbook you have sections, 2.1.1, 2.1.2. So break up your assignments into manageable pieces and get some of it done on one day, some of it another day. Don't leave it until Sunday night, or you will be scrambling around trying to get everything done. Okay, with that being said, please contact me, m.sigmon@snhu.edu. If you have any questions, if you're running into any trouble, if you just want to talk, that's fine. Email me, please. I'm here for you. I'm here to help you, okay? So please do let me know. And with that being said, I hope that you have a wonderful week this week.